Good morning everyone. In the last lecture we have seen the development of udder from birth to puberty and during the stress cycle, how the development of udder occurs during pregnancy and during lactation. Then we have seen the anatomy of cow udder. In that we have seen the location of pelvis, connective tissue, lateral suspensory ligament, left part of the udder, front part, right and rear part of the udder, median suspensory ligament. Then we have seen the functions of the suspensory ligaments, that is how it supports the udder and how the right and left halves are separated by a intramammary groove. Then we have seen the gland systems, teat systems, annular folds, parenchymal tissue, intermammary groove, skin of the mammary gland and the median suspensory ligament. Then we have seen the alveoli, what is the lobule, what is the ducts, major ducts, connective tissue, gland systems, teat systems and teat meatus. Then we have seen the internal structure of mammary gland. In that we have seen the glandular secretory tissues, connective tissues, ducts, gland systems, teat systems. And then we will see in detail the function of each part of the udder, that is a group of alveoli which are surrounded by a connective tissue septum forms a distinct unit called as a lobule. Lobule means a group of alveoli which is surrounded by a connective tissue septum which forms a distinct unit. That unit we call as a lobule, and a group of lobules are surrounded by a large amount of connective tissue forms a lobe, and the lobule empty into a small duct called the intralobular duct within the lobule, and it drains into a central collecting space from which the interlobular duct emerges. So within the lobe, the interlobular ducts unite to form a single intralobar duct. The interlobar duct may enter into the gland systems directly, or it may join with one or more other interlobar duct before entering into the gland systems. So in this way, the more amount of milk is stored in the ducts than in the cisterns. Then contractile cells means myoepithelial cells, also called as a basket cells, which resembles the smooth muscle fibers surrounded by a alveoli and ducts. And on contraction of these cells, the milk is let down from the alveoli and the ducts. Approximately 11% of the mammary development occurs before pregnancy and 41% during pregnancy, and the remainder takes place during early lactation. Then we will discuss about the surface anatomy of the bovine udder. The appearance of the udder varies depending upon the maturity and the functional status of the udder. In dairy cows, udder is very large and it can weigh up to 60 kg. The udder is divided into two halves. Sorry for the interruption. The udder is divided into two halves, each half having two quarters. Thus 4 quarters corresponds to the 4 glands, and each gland bearing a principal teats. Means a udder having 4 teats. Okay, and it is divided into two halves, and each half is having 2 quarters, and the 4 quarters correspond to the 4 glands, and each glands having a principal teat. Means in this way the udder is having a 4 teats. A median intermammary groove marks the division of the udder into right and left halves. The division between fore and hind quarters is less distinct. Then in short we will see the circulatory system of the udder. In this circulatory system, the caudal aorta gives off right and left common iliac arteries that branch to external iliac and pudic arteries. The latter gives rise to a cranial and caudal mammary arteries to supply the cranial and caudal part of the udder respectively. The major drainage route from the udder follows the same course as arteries from mammary veins to external pudic arteries and to the external iliac veins that joins to the caudal vena cava. Then another potential drainage route from udder is through the subcutaneous abdominal veins to the cranial vena cava. In lactating animals, a large quantity of blood is drained via this route. The dairy cow, the ratio of volume of blood flow through the mammary gland to volume of milk produced is about the 670:1. Means from the 670 ml of blood, the cow can form 1 ml of milk. Okay. So here in this diagram you can see how the circulatory system works. This is the subcutaneous abdominal vein. This is the external iliac artery. This is the inguinal canal, external pudic artery, and this is the perineal vein, and this is the papillary veins. So this is in short, and this is the vena cava, which drains drains blood from all the veins to the heart, and then it pures the blood, get purified, and then from the subcutaneous abdominal vein the blood is drained to the udder, and which forms the milk. Then we will discuss about the let down of milk. The udder, especially the teats, is stimulated by calf or milker. So the cows, especially the desi cows, requires the stimulation from the calf or from the milker at the time of milking, and this stimulation gives the impulses, and that impulses conducted along the nerves to the posterior pituitary at the base of cow's brains. Okay, that impulses are conducted through the nerves to the posterior at the base of posterior pituitary of the cow's brain, and that posterior pituitary stores and releases the hormone called as oxytocin into the bloodstream, and that blood transport oxytocin to the udder, and the released oxytocin causes the smooth muscles like cell surrounding each alveolus to contract, thereby forcing the milk out of them into a large duct and the cisterns of the udder, and that released oxytocin causes the contraction of the smooth muscles which are surrounded to each alveolus of the udder. Once the stimulation is obtained, the oxytocin released makes the milk let down and initiation, and it is very important the milk is removed within the time approximately 5 to 7 minutes to obtain greatest demand. Okay, milk can be removed within 5 to 7 minutes because the oxytocin released in that time only, and we can obtain the milk in 5 to 7 minutes only for greatest demand. So here we can see how the sight of cows give the stimulus, and sound of milking machines stimulates, and through the nerve impulse, and this nerve impulse goes to the posterior pituitary, and that posterior pituitary releases oxytocin in the blood, and that oxytocin contracts the smooth muscles of the udder, which is surrounds the alveoli, and that contraction gives the milk, initiates the milk, ejects the milk within 5 to 7 minutes. And even the touch of the milker to the udders, it also gives the stimulus for the release of oxytocin, and due to oxytocin milk ejection takes place.